Hi everybody, and welcome back to our Chapter 4 discussion of hormone control during exercise. To start our discussion, we need to talk about the endocrine system. The endocrine system functions as a communication system of the body. Our nervous system, from our previous chapter discussion, focuses on electrical communication. Our endocrine system, on the other hand, focuses on chemical communication. Due to its chemical communication nature, the endocrine system is much slower to respond but has longer-lasting effects than the nervous system. Our endocrine system also functions to maintain homeostasis via hormones. This helps us control and regulate cell and organ activity in the body and acts upon target cells. Our endocrine system also constantly monitors the internal environment and coordinates the integration of physiological systems at rest and during exercise. When exercising, homeostasis is maintained, as well as substrate metabolism. Our endocrine system also regulates fluid and electrolyte balance. Looking specifically at the two distinct types of hormones in our endocrine system, the first one are steroid hormones. Our steroid hormones will be derived from cholesterol. This means that they are lipid-soluble and can readily diffuse through cell membranes. Our steroid hormones are secreted by four major glands: the Adrenal Cortex, which is over the kidneys, which will release cortisol and aldosterone, the ovaries, which will produce estrogen and progesterone, the testes, which will produce testosterone, and the placenta, which is present in pregnant women, will also produce progesterone and estrogen. Our non-steroidal hormones are not lipid-soluble, which means they cannot readily cross cell membranes. This means that they will need assistance to cross the membrane. Our non-steroidal hormones consist in two groups. The first are protein or peptide hormones. These tend to be the most common non-steroid hormones, and these will be released from the pancreas, hypothalamus, and pituitary glands. We also have amino acid-derived hormones, which are mostly our thyroid hormones of T3 and T4, and our adrenal medulla hormones of epinephrine and norepinephrine. Hormones are secreted in bursts called pustules. This allows plasma concentrations to fluctuate over minutes or hours. We also have concentrations that can fluctuate over days and weeks. But what actually triggers and regulates these hormone bursts? This is going to be controlled by most things in the body by negative feedback. A hormone will be released which causes a change in the body. There will be large downstream changes that are going to reduce secretion. We will then have small downstream changes that will increase secretion. A good example of this is a home thermostat. When it starts to get warm in your house, the AC kicks on. Once it reaches the desired temperature, the AC kicks off. This is a negative feedback loop, and this is how hormones typically work in the body. Hormones are going to act in the human body in several different ways. However, because they are produced and released in bursts, the plasma concentration is a poor indicator of hormone activity in the body. That is because not only can we change the amount of hormones in the human body, we can also change a cell's sensitivity to hormones. We can also change the number of receptors on a cell's surface. The way we can control hormones is either through downregulation or upregulation. Downregulation is when we decrease the number of receptors during a high plasma concentration. This means we have become desensitized to that particular hormone. An example of a disorder from downregulation would be metabolic sensitivity decreases to insulin. So this is type 2 diabetes. Upregulation is when we increase the number of receptors during high plasma concentration. This is called sensitization. So, for example, when individuals complete high amounts of strength training that are type 2 diabetics, it increases their sensitivity to insulin, and therefore they can help bring glucose into their cells more effectively. Hormone receptors will also limit the scope of their effects because of our hormone-specific receptors. So even though hormones are released into the body and they circulate through the bloodstream, they can only act on cells that have the hormone-specific receptors. That means if there is no receptor on the cell surface, there can be no hormone effect. That means hormones can only affect tissues with their specific receptor. Hormones can then also only exert their effects after they have bound with that receptor. A typical cell that is going to interact with a specific hormone will have somewhere between 2,000 and 10,000 receptors. Once the hormone binds to the receptor, it creates something called the hormone receptor complex. Here's an example of steroid hormones in action. So, we know our steroid hormones are lipid soluble, so they can cross the cell membranes. We also know that steroid hormone receptors are going to be found inside the cell, either in the cytoplasm or the nucleus. Once the hormone binds to the receptor, it will enter the cell and travel towards the nucleus. This hormone will then bind to the DNA and direct any gene activations. This will regulate mRNA synthesis and protein synthesis. Steroid hormones can also enter a cell and have that hormone bind to a specific receptor in the cytoplasm or the nucleus. This hormone receptor complex will activate the cell's DNA, which will form mRNA. That mRNA will then leave the nucleus and enter the cytoplasm. The mRNA can then direct protein synthesis in the cytoplasm. This means that steroid hormones directly act within the cell to modify protein synthesis and the formation of organelles in the cell. On the other hand, if you remember, nonsteroid hormones cannot directly enter the cell because they are not lipid soluble. This means that they need to work with secondary messengers. That means the hormone will interact with a receptor on the cell and activate a secondary messenger chain reaction. These secondary messengers will be what actually carries out the hormone effects. The interesting thing about using our secondary messengers is it actually allows us to intensify the strength of a hormone signal. Our common secondary messengers are going to be cyclic androstenone monophosphate, or CAMP, or CAMP, cyclic guanine monophosphate, or CGMP, and isobutyl triphosphate IP3 and diaglycerol, or DAG. These nonsteroid hormones, as I've said before, cannot pass through the cell membrane. This means the hormone will bind to its specific receptor site on the cell membrane, and then the hormone receptor complex will activate an adenylate cyclase, or an ACMP, within the cell membrane. This adenylate cyclokinase, will catalyze to form CAMP. The CAMP will then activate protein kinases and other enzymes in the cell that will lead to cellular changes and hormonal effects. This is a positive cascade event. So, now let's look at our hormones more specifically. The first one will be our prostaglandins. These are going to form a third class of what we refer to as pseudohormones. They are going to be derived specifically from arachidonic acid, and these are going to act as local hormones. This means they will only work in the immediate area, so they are not true hormones. These mostly are going to work to mediate inflammatory responses like swelling and vasodilation, and sensitize our free nerve endings, or to tell us about pain in the particular area. Looking more closely at our specific endocrine glands and their hormones, we know that each of the body's several endocrine glands may produce more than one type of hormone, and then these hormones will regulate any physiological variables during exercise. Looking more specifically at hormonal regulation of metabolism during exercise, we need to be acutely aware of 4 specific endocrine glands and their responsibility in metabolic regulation. We need to pay attention to the anterior pituitary gland, the thyroid gland, the adrenal gland, and the pancreas. The hormones released by these glands are going to impact metabolism of carbohydrates and fats during exercise. We will start with the anterior pituitary gland. The anterior pituitary gland is going to be attached to the inferior surface of the hypothalamus in the brain. It will have 3 lobes, the anterior, intermediate, and posterior lobes. We are mostly focused on the anterior lobe in this module. The anterior lobe is going to secrete hormones that are going to respond to the hypothalamic hormone factors. This will include both releasing factors and inhibiting factors. Exercise is going to overall increase the secretion of all of our anterior pituitary hormones. Anterior pituitary gland is going to release a few different hormones, the first being growth hormone. Our growth hormone is going to be a potent anabolic hormone. This means that it is a hormone that works to build up tissues in the body. It also can improve the functionality and the size of organs. Growth hormone will also promote muscle growth, also known as hypertrophy, and stimulate fat metabolism. Our growth hormone release is going to be proportional to exercise intensity. This means that the higher the intensity of the activity, the more growth hormone that will be released. The next one we are concerned with is our thyroid gland. This is going to secrete triorthodoxorone and thyroxorone, or T3 and T4. T3 and T4 have a couple different impacts on the human body. They mostly lead to increases in metabolic rates of all of our tissues, protein synthesis, the number and size of mitochondria in our cells and muscle tissues, glucose uptake by cells, the rates of glycolysis and gluconeogenesis, or the breakdown of carbohydrates and the production of new sugars, and free fatty acid metabolism. Our thyroid gland will also be impacted by our anterior pituitary gland. Our anterior pituitary gland will release thyrotropin, also known as thyroid stimulating hormone, or TSH. This will travel to the thyroid and stimulate the production of T3 and T4. Exercise will also impact and increase the release of TSH. This means in the short term, T4 can increase after a delayed release. And if we participate in a prolonged exercise program, T4 will remain constant while T3 decreases. The next endocrine organ we are concerned with is the adrenal medulla. This is going to release our catecholamines. These are our fight or flight hormones. These will be epinephrine, which is about 80% of what is released, and norepinephrine, which is about 20% of what is released. When the adrenal medulla releases catecholamines, it will increase during exercise, which means we will see an increase in the sympathetic nervous system activation. This will lead to an increase in epinephrine and norepinephrine. Once catecholamine release is increased, this means we will have increases in our heart rate, contractile force, and systolic blood pressure. We'll also see an increase in the breakdown of glycogenolysis, or the breakdown of glycogen in the body, and the breakdown of free fatty acids. We'll also see an increase in blood flow to our skeletal muscle. Looking specifically at the adrenal cortex, this is going to release corticosteroids. This will also release glucocorticoids, as well as mineralocorticoids and gonadotropins. The major glucocorticoid we are concerned with exercise is cortisol. This is because cortisol is going to stimulate gluconeogenesis, the production of new sugars and carbohydrates. Cortisol can also increase free fatty acid mobilization and protein catabolism, or protein breakdown. Cortisol is important because it acts as an anti-inflammatory and depresses our anti-immune reactions. You can think of cortisol as a stress hormone. Next, we'll look at the pancreas. Mostly with the pancreas, we are concerned with insulin and glucagon. Insulin is going to work to lower blood glucose. This is going to counter hyperglycemia, or high blood sugar, and oppose glucagon. This will facilitate glucose transport into the cells. You can think of insulin as a key that unlocks the cells to allow glucose in. Insulin can also enhance the synthesis of glycogen, proteins, and fats. It will inhibit gluconeogenesis, or the production of new carbohydrates and sugars. Glucagon will work counter to insulin, and its job is to raise blood sugar, or blood glucose. This is going to counter hypoglycemia, or low blood sugar, and will oppose insulin. Glucagon is mostly going to work to promote glycogenolysis, or the breakdown of sugars, and gluconeogenesis, the production of new sugars. Next, let's look at the regulation of carbohydrate metabolism during exercise. So we know from our previous discussions that glucose must be available to our tissues, whether it is in an anaerobic or an aerobic type of activity. To do this, we need to convert glycogen to glucose for glycogenolysis, or we need to use the circulating blood sugars in our body. We can also produce new sugars called gluconeogenesis through free fatty acids or proteins and converting them to glucose. Having adequate glucose during exercise is going to require glucose to be released from the liver and glucose uptake by the muscles. Some hormones that can increase circulating glucose are glucagon, epinephrine, norepinephrine, and cortisol. Our circulating glucose during exercise will also be affected by growth hormone, which allows free fatty acid mobilization and cellular glucose uptake, as well as T3 and T4, which is going to impact glucose catabolism and fat metabolism. The amount of glucose released from the liver will depend on the exercise intensity and duration. Next, we know as exercise intensity increases, catecholamine release will increase, and glycogenolysis rates will increase, so liver and muscles will start to break down glycogen. This also means muscle glycogen will be used before liver glycogen. As our exercise duration increases, more liver glycogen will be used, so we'll have an increase in muscle glucose uptake and an increase in liver glucose release. This also means that our glycogen stores are going to start to decline, and our glucagon levels are going to start to climb. But glucose mobilization and getting it to the cells that need it is only half of the story. We need to actually be able to get glucose into the cells. This is where insulin comes into play. Insulin is what enables glucose uptake into our working muscles. This means during exercise, our insulin concentrations will decrease. This will allow cellular insulin sensitivity to increase, and more glucose can be taken into the cells, and therefore less insulin is used. Here is an example of hormone levels and how they are going to fluctuate during a bout of physical activity. So you can see epinephrine will be in yellow, norepinephrine will be in red, glucagon will be in green, cortisol will be in purple, and glucose will be in blue. You notice that during the bout of physical activity, glucose levels remain fairly constant. However, how we get those glucose levels to remain constant is constantly going to change. You notice early on in physical activity, you will see a big spike in cortisol, which will then start to decline. You will also see a rapid spike in glucagon, which will then start to level out, and a steady climb in epinephrine and norepinephrine. I want you to take a moment and kind of think of how these hormones are going to interact with each other to tell the story of how physical activity and glucose needs are going to change during the bout of activity. Next, we need to talk about how the central nervous system and the endocrine system are going to interact. The central nervous system will regulate carbohydrate metabolism through the hormones, specifically insulin, and the nutrients, basically glucose, fatty acids, and amino acids. The brain is going to be sensitive to glucose and is going to help control insulin release. We also know leptin and GLP-1, which are hormones released by adipose tissue and the gut, are going to act through the central nervous system to decrease glucose production. This is very important because glucose is the only substrate that the brain will use for metabolism. This means if you do not have adequate carbohydrate intake, even during physical activity, you will start to starve your brain of resources and substrates to use for energy production. This is why when individuals go on low carbohydrate diets, one of the side effects tends to be general mood swings and crankiness. Your brain is very upset that you are not feeding it. So, as we can see here, this is an interaction between glucose and insulin levels. Try to think back to what we just discussed, and I want you to take a moment to explain this kind of table to yourself. I feel like this would be a good test question. Hint, hint, wink, wink. Next, we need to discuss the regulation of fat metabolism during exercise. So, free fatty acid mobilization and fat metabolism are going to be critical to endurance exercise. The longer we participate in physical activity, the more free fatty acids we will need to use. This is because glycogen will become depleted at some point, so fat energy substrates will be needed to supplement substrate supply. Therefore, we need to break down fats through lipolysis, and this will be accelerated. To start, we will take triglycerides and break them down into their component parts of free fatty acids and glycerol. This is because fat is stored as triglycerides in our adipose tissue in our body. Once we have broken down into free fatty acids, we can transport these free fatty acids to the muscle. The rate of triglyceride breakdown into free fatty acids is a possible determinant of the rate of cellular fat metabolism. If you are efficient at breaking down triglycerides, you'll be able to get the free fatty acids to the muscles faster. Lipolysis will be stimulated by a decrease in insulin, epinephrine, norepinephrine, cortisol, and growth hormone. They are all going to stimulate lipolysis via the enzyme lipase. Additionally, hormones in the body are going to regulate fluid and electrolytes during exercise. This is because during exercise, plasma volume is going to decrease. This is going to cause an increase in hydrostatic pressure and an increase in tissue osmotic pressure. We'll also see a decrease in plasma water content due to sweating, and an increase in heart strain and a decrease in blood pressure. Hormones that are going to help correct our fluid imbalances will be the posterior pituitary gland, the adrenal cortex, and the kidneys. Looking more specifically, let's talk about the posterior pituitary gland. The posterior pituitary gland is going to secrete an antidiuretic hormone, or ADH, and oxytocin. These are going to be produced in the hypothalamus and then travel down to the posterior pituitary. These will then be secreted upon the neural signal from the hypothalamus. Only ADH will be involved with exercise. ADH will function to increase water reabsorption at the kidneys and make us have less water in our urine. Therefore, it is an antidiuretic. This is why when you start to participate in physical activity, you can get dehydrated and your urine color becomes darker because there's less water in it. The posterior pituitary gland will also serve as stimuli for ADH release. This means when we have a decrease in plasma volume, we will have a change in the hemo concentration, or the concentration of our blood, which will increase our osmolality. When we have this increase in osmolality, this will stimulate the osmoreceptors in the hypothalamus to release ADH. ADH will then be released, which will increase water retention by the kidneys. This allows us to have a minimization of water loss and prevent severe dehydration. Looking at this figure, we can see that muscular activity will promote sweating. The sweating will cause a loss in our blood plasma, resulting in a change in our hemo concentration and an increase in blood osmolality. This increase in blood osmolality will stimulate osmoreceptors in the hypothalamus. The hypothalamus will then send a neural signal to the posterior pituitary gland. The posterior pituitary gland will then secrete ADH into the blood. ADH will act on the kidneys, which will increase the water permeability of the renal tubules and the collecting ducts in the kidneys, leading to an increase in the reabsorption of water. Decreased urine output will aid in minimizing blood fluid losses. Next, let's talk about the adrenal cortex. The adrenal cortex is going to secrete minimal corticoids. A major mineralocorticoid is aldosterone. Aldosterone will impact the body by increasing sodium ion retention by the kidneys. This will also lead to an increase in water retention via osmosis and also an increase in potassium excretion. What stimulates aldosterone release is a decrease in plasma sodium ion concentrations, a decrease in blood volume and blood pressure, and an increase in plasma potassium concentrations. There is also an indirect stimulation by a decrease in blood volume and a decrease in blood pressure in the kidneys. Next, let's talk about the kidneys. The kidneys are going to be the target tissues for ADH and aldosterone. Our kidneys also secrete a substance called erythropoietin, or EPO, and renin. EPO will be released in response to low blood oxygen in the kidneys. This will stimulate red blood cell production. This will be critical for adapting to training, and specifically training at altitude. This is why EPO is a very popular blood doping kind of cheating substance. Also, the stimulus for renin, which is a particular enzyme in the kidneys, is when we have a decrease in blood volume and a decrease in blood pressure. This is going to stimulate the sympathetic nervous system via impulses. This will activate the renin-angiotensin-aldosterone mechanism. This means renin will convert angiotensin to angiotensin 1. We will then take ACE to convert angiotensin 1 to angiotensin 2. And then angiotensin 2 will stimulate the aldosterone release. So you can think of this as a cascade effect. Looking at this particular model, I'll walk you through this mechanism. So, looking with prolonged exercise without adequate fluid replacement will lead to dehydration. Dehydration can then in turn cause a decrease in blood pressure, which will be sensed by the kidneys. Renin will then be secreted by the kidneys. Renin will then convert the protein angiotensin, which will be released by the liver, to angiotensin 1. Angiotensin will be a converting enzyme in the lungs, converts angiotensin 1 to angiotensin 2. Then angiotensin 2 will stimulate the adrenal cortex to release aldosterone, and it will vasoconstrict blood vessels, which will increase blood pressure. Aldosterone will then act on the kidneys to increase sodium ion reabsorption. This graph shows the interaction between plasma volume and aldosterone concentrations. As you can see, as physical activity continues over time, plasma volume will start to decrease. This will cause an increase in aldosterone to try to minimize the change in plasma volume. Finally, in our conversation of fluid and electrolytes, we need to discuss osmolality. Osmolality is the measure of concentration of dissolved particles, particularly proteins and ions, in the body fluid compartments. The normal osmolality values is somewhere between 300 milliosmoles per kilogram. Osmolality and osmosis are going to interact. So if a compartment osmolality increases, this means water will be drawn in. If compartment osmolality decreases, that means water is drawn out. Aldosterone is going to impact osmosis. If we have sodium ion retention, this will lead to an increase in osmolality. This means water will be drawn in. This is why if you eat a particularly salty meal, you tend to hold on to water. When you have an increase in osmolality, this will lead to an increase in water retention. So, where sodium moves, water will follow. This means osmotic water movement will minimize the loss of plasma volume and maintain blood pressure. Additionally, ADH-aldosterone effects are going to persist for somewhere between 12 and 48 hours after exercise. Prolonged sodium retention can lead to abnormally high sodium ion concentrations after exercise. Since we know that water follows sodium, prolonged rehydration effects can occur if adequate sodium ions are consumed. This is an example of real-life sodium retention. It is fairly common in the Highland Games that I participate in for it to be very, very hot in the field, somewhere between 100 to 110 degrees, especially during the summer. It is a very common practice to consume Gatorade or Pedialyte, but another common practice is to actually consume salt with your water. There are many Highland Games athletes that I have seen quite literally put pink Himalayan salt into their hands, lick it off their hand, and then chug a bottle of water. Well, we know when you consume those high levels of sodium ions, this is going to allow water to fill the cells. This means that you will actually stay hydrated longer. Our final point of discussion will be hormonal regulation of calorie intake. The hypothalamus is our brain's appetite control center. Satiety center is going to be in the ventromedial nucleus, and our hunger center is going to be in the lateral hypothalamus. This means that our hypothalamus is going to control whether we're hungry or we're satiated or full. Our GI tract is also going to release hormones that affect our hunger signals. CCK will be stimulated when the stomach is full. This will decrease appetite. This is why you don't want to scarf your food. You need to give time for CCK to tell your brain that you're full. We also have glucagon-like peptide 1 or GLP-1. This will be released in the small intestine, which will help us decrease appetite. We also have peptide YY or PYY, which will be released in the small intestine, which will decrease appetite. And then we have ghrelin, which is going to increase our appetite. Additionally, adipose tissue in the body acts as an endocrine organ. Leptin will be released from the adipose stores and will reduce hunger. Leptin and ghrelin will act in opposing ways. Individuals that are obese can have higher leptin but are often resistant to its effects. Exercise, however, can affect hunger and satiety hormones. This means that acute, vigorous exercise can increase PPY and GLP-1, therefore reducing hunger. Exercise training, however, does not affect a ghrelin except in a particular energy deficit. Thank you guys for listening to our discussion of Chapter 4, and I look forward to hearing from you guys again for Chapter 5.